So I would like to introduce Professor Angelo Lombardo from also Italy, from the same institute as Chiara. He's also quite well known, published quite a bit in gene editing. He did his training with Professor Naldini at the beginning, but has risen quite to the star. And we'll talk about other ways of editing, particularly also silencing gene expression in cells that are of interest.
Thank you. So thank you very much for the introduction and for the invitation. It's really a pleasure to be here, and really hope actually to learn much about this field. So as said, I will mostly discuss about this quite novel approach of editing cells, which is based on the modifications of the epigenome. So this is my conflict of interest. And I have actually subdivided the presentation in 3 parts. So a background on the epigenome strategies, which might not be familiar to you. And then I'd like to share with you some projects which are ongoing in the lab. One of them actually relates to engineering or multiple engineering of T cells for cancer immunotherapy. So let's start by discussing the editors that we have available. And Chiara already did a very nice introduction on what we can do with these multi-phased technologies, which actually are around since more than a decade. And it's probably obvious to this audience that we are now available, we can now really modify in a precise manner our genome, cleaving the genome, as Chiara said, using nucleases of different kind, nicking the genome, or really simply swapping bases or changing bases into the genome. I've been in the field of editing, again, since many times. What I always find also fascinating is the fact that we can do these things thanks to the fact that we do have available a wide range of DNA binding technology that we can really program to target our sequence of interest. And I will discuss some of them in my talk. So Chiara mentioned zinc finger proteins. Said, not easy to be designed, but very proficient. Transcription-like effectors, very similar DNA binding, let's say, approach. And the recent comers, which are the CRISPR-Cas9 technology, that can be used very rapidly and effectively by many of us. On this side, we can really harness and exploit this DNA binding domain to tether on the genome other effector proteins. Base editing is one example. All other kind of effector proteins which may actually modulate gene expression. For instance, repressing gene transiently or permanently, or activating gene. This is known as epigenome editing. What is epigenome? In a very simple fashion, it's a field which refers to any kind of changes in the chromosome that are affecting gene activity and expression, not really, though, impacting on the primary DNA sequence. And you can see that in 2 ways. So you may have chemical modifications in the histone tails that allows the gene to be expressed or accessible to transcription factor or expressed. Other different modifications on the histone tails, and I will talk about that. Also DNA methylation, which on the other hand may compact the genome and results in gene repression. So what are the players in the game we are doing? So these are epigenome editors, which are very similar to the technology that just Chiara mentioned. So you have a DNA binding domain. You can program this DNA binding domain towards your sequence of interest, and you fuse this DNA binding domain to effector domains of different kind, most of which actually taken from naturally occurring proteins which modify actually the epigenome. And there are different ways by which you can do epigenome editing. You can impose histone modifications, which may be either activatory or repressive. P300 is a well-known activator of gene expression. This domain that we'll discuss later, the CRAB domain, is a famous repressor of gene expression. And on the other side, you can play with the methylome of the DNA, so imposing new DNA methylation or promoting the demethylation of this DNA. So imposing methylations implies gene repression. Removing this methylation implies gene activation. Just give you an example of that. So in this scheme, you can see an editor, an epigenome editor. So this is the DNA binding domain of the editor, which has been fused to the CRAB domain, which, as mentioned, is a powerful repressor. You can transiently express this editor into a cell. Thanks to its DNA binding domain, it recognizes the intended target sequence, rewrite the epigenetic of the locus towards a repressive state, and silence a gene. That's a very powerful approach. However, when the repressor is gone, the gene tends to reactivate. So it's a nice approach by which actually you can promote robust and transient waves of gene repression. Here there is an example. So this is Pt.2m, the Pt.2m gene, the architecture of the Pt.2m gene. You can program the editor to recognize the promoter, and this is the outcome of that. So it's very powerful, but as you can see here, you need to actually express the repressor for a long period of time. Otherwise, the modifications will be removed and diluted. So a few years ago, actually, we were inspired by this beautiful process which occurred early during development, by which actually endogenous retroelements or retroviruses are permanently silenced through epigenetic mechanism in our cells. So this process contains, let's say, two pathways. The first pathway is still a pathway in which there are these CRAB proteins, zinc finger proteins, which recognize specifically the sequences of these endogenous retroviruses early during development. And you can see from this cartoon, this scaffold domain recruits a plethora of other epigenetic factors which modify the histone tails toward a repressive state. These proteins, though, recruits also these other classes of molecules, DNA methyltransferases, which then methylates the DNA, finally locking this repressive state in a cell. So why it's so intriguing this? Because, again, in this pathway that is encoded, let's say, the inheritance of epigenetic modification. And you can use that, actually, now to program permanent gene silencing. Here there is an example on how this pathway is working, a schematic of how this pathway is working. Again, very early during development, this pathway is active when the endogenous elements are expressed, and then they are off, and they remain off for the entire life of the individual. So we were inspired by that and took some of these effectors to build an epigenetic editing platform that may allow you to permanently silence genes by transient expression of this molecule. And here there is an example. These are these cocktails of factors. We selected CRAB domain, the recruiter, and these two DNA methyltransferases. And you can see from the scheme, a transient expression of these proteins is enough to promote robust and durable silencing. As shown also here, we are again looking at Pt.2m. This is the CRAB that you've seen before. These are the activities of the other two effectors, 3L and 3A alone. But when you mix the three components together, you have a nice synergy among them and a nice levels of silence which remain stable during time. So that's the premises of the technology. People, we and others, have shown that it is very effective and specific and durable in cell line, and that DNA methylation is the driver of this inheritance of repression. It is reversible at will because you can either use drugs that may promote DNA demethylation to reactivate the gene or target activators that promote, again, DNA demethylation. How to translate this approach into clinics? So that's actually the goal of our activity in the lab. And here I would like to show, as I mentioned, a couple of examples on this. Well, the first one deals with polyfunctional editing or engineering of T cells. I don't really want to discuss much about that because I've been nicely introduced by Chiara, and I think the audience is well aware of the roadblocks which somehow limits cancer immunotherapy. What is probably clear to everybody is that having cells heavily engineered that may somehow ameliorate or potentiate the activity of these engineered cells. And what I'm referring to multiple engineering is that you can at the same time eventually program knockout, eventually program knock-in, on eventually modulate expression of genes in a single hit. And the literature is telling us that there are now many genes that we can now modify simultaneously in these engineered cells. And one example of these multiplex editing comes from clinical trials. And a beautiful example is this study in which actually they use gene editing with the CRISPR-Cas9 to disrupt the TRAC locus, the PD1 locus, and insert into these edited cells a novel TCR. So these studies also teach us that when you do multiplex editing, you may risk of having translocations among the multiple edits you have in the cell. And here is the example in which they also disrupt Pt.2. When you do that, the cells repair the break also by having translocation among the different breaks. That is it's a challenge, it's a problem when using these editing technology in clinic. So how to maybe solve this issue? Well, first of all, you can use this epi silencing platform to perform safe multiplex gene silencing. And we actually started by testing epi-silencing of a Pt.2, which is a target that may be used actually to prevent rejection of the engineered cells while transplanting in an allogeneic setting. And that was not easy, actually, but at the end of the day, we came out with a protocol which allow us to achieve a very nice epi-silencing I'm going to show you in a minute. So here we are basically transfecting RNAs and coding for the epi editors in the T cells. So again, it's a hit and run approach. So these editors are expressed for a very short time in the cell, and then we monitor during time efficiency of silencing of this specific gene, which is Pt.2. So this is an example. So I'm showing here the percentage of Pt.2 negative cells over time, over 2 rounds of restimulation of the cell. In this specific case, we are using actually 2 different architectures of editors, but it's not so much relevant here. But as you can clearly see, very rapidly in 95% or more of the cells, the Pt.2 gene was lost. So you can see also here in this flow cytometry analysis, here are the few remaining Pt.2 positive cells, and most of the cells actually basically lost expression of Pt.2. And it's quite robust, so we use that technology in many different blood donors obtaining significant efficiency of silencing. That was the first gene, and again, we set the technology. I'm not detailing much what we did here to make this platform work in 100%. Was difficult, but at the end we did it. And then we asked, can we also silence other genes in these cells by epi-silencing? On that, we focus on 2 genes, TGF-beta receptor 2 and Pt.2. The first one have been proposed actually to boost CAR T cell activity against solid tumor, and the second one has been proposed to increase persistence of T cell, CAR T cells in patients. And again, here we are using these the ETRs targeting the promoter of these two genes. And in this specific experiment, actually, we basically tilled the entire promoter of the gene, searching for the most effective guide RNA, because all these experiments were done with Cas9 ETRs. And you can see here from these maps, we can, let's say, identify very effective guide RNA to silence both genes. So what we basically established in the beta-2M was reproducible also in these two other genes. So the question now would be, can we multiplex the technology so can simultaneously silence epigenetically 2 or 3 of these genes in T cells, so human primary T cells. It is an example in which basically we program the editors to recognize the beta-2M promoter or the Pt.2 promoter. The flow cytometry showed you that beta-2M was nicely silenced in these cells, 91% of the cells with negative beta-2M, stable in time in culture. Digital droplet PCR to measure the transcript of beta-2M and to confirm silencing of beta-2M, and uncover the fact that also Pt2 was nicely silenced in these cells. As mentioned, so one limitation of editing, especially gene editing in these cells, or in general when you do multiplexing, is the fact that by concurrent disruption of the two genes you may have translocation, and this is indeed the case. It is a really qualitative analysis. We are trying now to quantify all of this, but if you do the multiplex editing with the standard Cas9, so gene disruption, you do see, this is an example, translocation between the beta-2M and Pt2. As these technologies, so epi-editing, lacks DNA cleavage activity, you do not see that. So you do not have translocation between the two genes. This was the first, let's say, multiplexing experiment we did. Then we challenged the technology, trying to silence simultaneously the 3 genes, so beta-2M, Pt2, and TGF-beta receptor, using different architecture. So there is room for improvement here. As you can see, from 50 to 70% of the cells were beta-2M negative, according to the fusion we are using. And we can nicely see multiplex silencing. And also in these cases, when we inspect for translocation, we do not find translocation when using this epi-editing technology. So let me then move to the second part, because as I mentioned, orthogonal edits might be very relevant to engineer T cells. And what I mean with orthogonal edit is not only silencing a gene, but at the same time eventually edit another locus, for instance, by targeting insertion, as Chiara has shown. So at the same time we were developing this multiplexing approach, we also thought to develop a polyfunctional editing platform that allows you to perform this orthogonal editing. And we named this new technology polyeditor. So let me go back to the scheme in which we can discuss about what happens when you do multiple gene editing in a cell. So as mentioned, so you can induce translocation between the two cleavage sites. And if you want to perform these orthogonal edits using these ETRs, which I will remember are not endowed with catalytic activity, you will end up anyway having translocations, because the guide RNA we are using, I apologize, I'm quite technical here, but the guide RNA we are using here are not really orthogonal. So if you want to silence beta-2M with the ETR and at the same time disrupt or insert something in another gene, by using such guide RNA of equal length, you will also program the Cas9 to cleave the gene that you want to silence epigenetically. So how can we solve this problem and make really a technology which at the same time can promote, for instance, HDR, so insertion of transgenes, and silence a gene without causing translocation by using guide RNA of different length? It's been shown by many many laboratories, that truncation of guide RNA inactivates the catalytic activity of Cas9. So you can now build a catalytically competent Cas9 repressor, okay? And then program the two outcomes, so gene editing, classic gene editing, and epigenetic silencing or gene activation by treating the cells with the guide RNA of different length. I hope that is clear. And in this specific case, having truncated guide RNA, you are not cleaving anymore the gene you want to silence, but you switch it off by epigenetics. So at the same time, you can have gene editing and epigenetic silencing, or gene activation, if you will. So very briefly, how to show you how this platform is working. Here we are independently testing the idea of using truncated guide RNA. So we use classic Cas9, cleavage competent, and epi editors with inactive Cas9, and then look at different guide of different length to see when the catalytic activity of Cas9 is disabled, and if the epigenetic silencing activity of the editors is still active at short guide RNA. You can see from here, by truncating guide RNA, you rapidly lose activity of Cas9. So with truncated guide RNA of 16 or 15 nucleotide long, you do not have any more cleavage of your target. At the same time, at the same length of guide RNA, you still have epigenetic silencing. So this is an experiment in which actually you use, again, this guide RNA of truncated length together with this poly editor, cleavage competent Cas9, fuse the effector domains that I mentioned to you before, to program epigenetic silencing of beta-2M, disruption of TRAC-C, at the same time insertion of these NY-ESO-specific ETR, as Chiara showed also before. So a single treatment led to the generation of 80% of beta-2M negative cells, not because of disruption of the gene, but because of epigenetic silencing of that. At the same time, the cells were roughly 63% NY-ESO positive because at the TRAC-C we are inserting the NY-ESO. Then if you inspect the expression of NY-ESO in beta-2M, you find that roughly 40% of the cells that are NY-ESO positive are actually beta-2M negative. So in a single step, you may achieve orthogonal edits in these cells, which are target integration on one side and epigenetic editing on the other side. And again, it's using qualitative analysis and comparing this approach in which we use guide RNA of different length, full length against TRAC-C or truncated against beta-2M, versus full length guide RNA targeting both beta-2M and TRAC-C with these catalytically active editors. We see that there are clear translocations among the two loci with a standard, let's say, gene editing, but that is absent in using this epi-editing approach. To conclude this part, I mean, we were able to, through, again, protocol optimization, and I want to go too much into the detail of that, but we were able to achieve efficient and durable silencing of multiple genes in T cells with epi-editing, so without chromosomal translocation. We then developed this poly-editing approach in which we can basically achieve at the same time in these cells both gene targeting or gene editing and epigenetic silencing. And as I mentioned, as I showed, without causing reciprocal chromosomal translocation among the genes that we are targeting. We are now evaluating the specificity of this treatment and their functionality in vitro and in vivo. So very briefly, let me move to the second part, which actually deals to the application of this technology in vivo and specifically in the liver. One of the most relevant questions here is how durable is epigenetic silencing once imposed into a living organism. So here we target this gene, which is named PCSK9. These are quite interesting and relevant genes for hypercholesterolemia because these PCSK9 is actually expressed by hepatocytes and binds to the LDL receptor, promoting actually lysosomal degradation. And there are nice examples in human in which loss of function mutations of these PCSK9 are protective against coronary heart disease. Why? Because the LDL receptor stays longer on the plasma membrane of the hepatocyte, capturing LDL and associated cholesterol from the blood of this subject. On the other side, there are gain-of-function mutations causing familiar hypercholesterolemia. So the first point is that Mother Nature tells us that you can actually inactivate this gene into the liver of subjects without causing much problems, actually protecting them from coronary heart disease. And indeed, this is a famous target in this field, and there are several approaches by which you can genetically or even epigenetically inactivate this gene. In the case of epigenetic inactivation, as I showed you at the beginning, people have used the CRAB domain to silence PCSK9, but as this domain is, the modification imposed by this domain are transient, people using stable expression of this repressor in hepatocytes, which is something that we don't want to do, actually. We want really to have a hit-and-run platform here. So I don't enter too much into the details, but just to tell you that at the beginning, we basically tested a different DNA binding technology, the CRISPR-Cas9, tail and zinc finger proteins, targeting the PCSK9 gene, the specific studies, the murine PCSK9 gene, searching for the most effective among these platforms. And through several rounds of iterative screens, actually, we were able to identify zinc finger proteins, which were actually very effective in silencing PCSK9, and don't think you can really see that, but at, let's say at low doses, the zinc fingers were working as efficiently as standard gene disruption with Cas9 at 10 to 6-fold higher efficiently than tail or dead Cas9 epi editors. So what about the specificity of these approaches? I haven't mentioned that much. It's, of course, a very relevant question. So once you express these epi editors in a cell, where these epigenetic modifications are imposed and how they are impacting on the transcriptome, for instance, of a cell. So here you can see RNA analysis or transcriptomic analysis of cells which have been exposed and heavily silenced with the epi editors targeting PCSK9. And nicely enough, you can see that the only downregulated gene in this specific experiment was PCSK9. Very comparable results with the gene disruption, as you can see here. So the transcriptomic wide approach tells us that the platform is really specific. A second thing that you can do to profile specificity of this platform is to look at DNA methylation, de novo DNA methylation, because as I mentioned, that's the core of the inheritance of this platform. So we perform a whole genome bisulfite analysis. Here we are exclusively looking at the PCSK9 gene in wild-type cells, in episilent cells, or in disrupted cells. You can see the differences in terms of CpG methylation, so de novo DNA methylation between epi-silencing and the other two approaches. At the genome-wide level, actually, we measure comparable hyper or hypomethylated sequences, let's say, regions, between the epigenetically silenced and the gene-disrupted cells, indicating that, again, that the procedure was not causing differences in terms of DNA methylation between controls and episilent cells. So we do have effective ETRs, so effective platform to silence PCSK9 specific. Now we need to deliver them to the liver. And we do have different vehicles to do that, so viral-derived vectors, first of all, but we don't like them much because, as I mentioned many times, you don't want to have permanent expression of these epi editors in your cells because that may be problematic. That may increase significantly, for instance, off-target activity of the proteins, which are actually chimeric proteins which so they may have maybe also recognized by the immune system as a foreign antigen. So here we reverted to the really famous now lipid nanoparticle technology. You can package in these particles messenger RNA encoding for your ETRs and achieve very nice transient expression of this molecule into the liver. We spend a lot of time to optimize the payload, so optimize the mRNA to be delivered. We perform screen to identify effectively lipid nanoparticles and selected one of them, and then perform an in vivo experiment in which, again, we injected mice with particles containing messenger RNA against PCSK9. And here you can see the data. So these in black are the curves of circulating PCSK9 in mock-treated or PBS-treated mice. This is instead what you can achieve by a single treatment of these with these particles containing the epi editor. So roughly 50%, you have a 50% decrease in terms of circulating PCSK9, indicating effective silencing of the gene in vivo. So again, now we have a longer follow-up. We are around 220 days after the treatment and still remain stable. So we can really impose efficient and stable gene silencing upon a single administration of DTR, even in vivo. And that's actually the conclusion of this story. We are still investigating the specificity of these treatments in vivo and evolving the architecture of the epi editors in order to further improve their activity. With that, I would like to close by thanking many talented people working in my lab, in particular Martino Alfredo Cappelluti, who is responsible for the PCSK9 project, Tania Baciaga, together with Deborah Cipria, who are responsible for the polyfunctional editing stories, and all the people really in the lab for fruitful discussion. Collaborators at Ospedale San Raffaele, in particular Luca Vago, for some of the studies we are doing with engineered T cells, and need you for your attention. Thank you very much.